Put your hand flat against your own arm right now and press down, just for a second, on the skin over the bone underneath. Feel how solid that is. Now think about this. Somewhere in Southeast Asia, in the wet, dripping dark of a limestone cave, there's a bone just like that one from a body that stopped moving 50,000 years ago. 50,000 years. Not 5,000, not 5,000. 50,000 years of rain, heat, insects, fungus, and time pressing down on it. And yet, when scientists finally opened that bone up in a lab, something inside it was still alive in a sense. Not literally alive, but intact enough to read. It's DNA. The molecule that once carried the instructions for building a human being was still sitting there, fragmented and fragile, waiting for someone with the right tools to come along and listen to what it had to say. And what it said didn't confirm the old story. It broke it. For most of the 20th century, the standard picture of human history was simple, almost tidy. Modern humans evolved in Africa, walked out sometime around 50 to 70,000 years ago, and spread across the world in one long wave, replacing whatever came before them. Southeast Asia was treated as a place people passed through on the way to Australia, a kind of hallway between the African homeland and the Pacific. Few people expected the hallway itself to be hiding one of the biggest surprises in the whole story. But that is exactly where the DNA and old bones revolution has landed some of its heaviest blows on what we thought we knew. And this video is about how that happened, what the bones actually revealed, and why the twist in the middle of this story caught even the specialists off guard. Before we get into the twist, we need to go back to where this kind of science actually started, because the story of ancient DNA is really a story about people who were told something was impossible and did it anyway. In the early 1980s, a small group of researchers working with old museum specimens tried something that most of their colleagues considered close to science fiction. They wanted to see if DNA, the molecule that carries genetic instructions, could survive death by decades or centuries instead of disappearing the moment an organism died. Their first big success came not from a human bone, but from a preserved animal skin, a quagga, a now extinct relative of the zebra, sitting in a museum drawer, when they managed to pull out and read tiny fragments of DNA from that old dried tissue. It proved something huge. Genetic material could survive far longer than anyone had assumed if the conditions were right. That single result cracked open an entire new field. Researchers who had spent careers studying bones by looking only at their shape and size suddenly had a new question in front of them. Could they read the genetic code buried inside those bones too, not just guess at relationships from how a skull was shaped? That question sat unresolved for years because the technology to actually do it cheaply and reliably did not fully exist yet. DNA does not want to survive. The moment an organism dies, ordinary chemical processes start chopping the long DNA molecule into smaller and smaller pieces, like a book left out in the rain until the pages turn to confetti. Bacteria move in and feed on what is left. Water breaks chemical bonds. Heat speeds all of this up dramatically. By the time a bone has been in the ground for even a few thousand years, the DNA inside it, if any survives at all, is usually reduced to short, damaged fragments, a few dozen genetic letters long instead of the billions found in a living cell. This is where Southeast Asia enters the story as its own kind of villain and its own kind of prize. If DNA hates rain, heat, and time, then Southeast Asia is close to the worst possible environment for it to survive. The region sits near the equator. Average temperatures often stay warm year-round. Humidity stays high. Many of the caves and river sites where ancient human bones turn up are damp for most of the year, sometimes flooding seasonally. Compare that to places like Siberia, Greenland, or the high mountains of Europe, where cold acts almost like a natural freezer, slowing down the chemical clock that destroys DNA. Researchers spent decades assuming that truly ancient human DNA, tens of thousands of years old, simply could not survive in a tropical climate like this. Southern and Eastern Asia became known, almost as a running joke among specialists, as a kind of DNA graveyard, full of bones, empty of genetic answers. That assumption is the tension at the center of this whole story, and it is worth sitting with it for a moment because it shaped textbooks and shaped what questions researchers even bothered asking. If you can't get DNA out of a region's oldest bones, you can't directly test who lived there, who they were related to, or whether they mixed with other human groups passing through. You are stuck working only with bone shape, tooth wear, and stone tools, which can tell you a lot but leave huge gaps. For a long time, entire university courses taught that reliable ancient human DNA, older than about 10 or 15,000 years, from a hot, humid environment, was basically off the table. Some early attempts to extract DNA from tropical fossils failed completely, or worse, picked up modern contamination, DNA from the people handling the bone, and mistakenly reported it as ancient. Those false starts hardened the assumption even further. If tropical ancient DNA science had a reputation, for years it was, don't bother. So how do you actually get real, ancient, uncontaminated DNA out of a bone that has spent 50,000 years in a hot, wet cave? This is where the mechanism of the science becomes almost as interesting as the results, and it helps to use an everyday comparison to understand it. Think of DNA as an incredibly long instruction manual, written in a 4-letter code, folded up tightly inside every cell of your body. In a living person, that manual is complete, readable start to finish. In an ancient bone, it is more like a manual that has been through a shredder, a flood, and a fire all at once. Most pages are gone. The ones that remain are torn into small scraps, sometimes only a sentence or two long, and some of those scraps have been chemically altered so that a few letters look different from how they originally read. The first stage of the mechanism is finding a part of the skeleton where DNA is most likely to have survived at all. Researchers learned, mostly through trial and error, that certain bones protect DNA far better than others. The dense, rock-hard bone found inside the ear, called the petrous bone, turned out to be a small miracle in this respect. It is one of the hardest, densest bones in the entire human body, almost like a tiny stone vault sitting inside the skull. Because it is so dense, it lets in less water and fewer bacteria than more porous bones like ribs or arm bones. Teeth, especially the root and the dense inner layer called dentine, turned out to be a second reliable vault for the same reason. Once researchers realized this, they stopped hunting through whatever bone happened to be handy and started specifically targeting these two structures whenever they wanted the best shot at ancient DNA, even in difficult tropical conditions. The second layer of the mechanism is even more delicate, the actual extraction. This has to happen in an ultra-clean lab, often in a separate building from any lab that works with modern human DNA, because modern human DNA is everywhere. It is on our skin, in our breath, floating in ordinary household dust. If even a tiny trace of a living researcher's own DNA gets into the sample, it can completely swamp the small amount of genuinely ancient DNA and make the whole result meaningless. So researchers work in full protective suits, masks, gloves, and hairnets in rooms filtered to remove stray DNA from the air, treating the ancient bone almost like a crime scene where any contamination ruins the evidence. They drill a small amount of bone or tooth powder under these sterile conditions, then use chemical processes to pull whatever fragmented DNA is present out of that powder and into a liquid solution. The third layer, and this is where modern computing enters the picture, is sequencing and reassembly. Because ancient DNA survives only in short, damaged fragments, researchers cannot just read it, start to finish, like a book. Instead, they generate millions of tiny overlapping fragments and feed them into powerful computers that compare each fragment to a reference genome. Essentially a giant, complete modern human instruction manual used as a map. The computer looks for fragments that overlap and stitches them back together, the same way you might reconstruct a shredded letter by matching torn edges and repeated words across scraps of paper. Special software also has to recognize telltale chemical damage patterns that build up over thousands of years, so scientists can tell the difference between this is genuinely ancient DNA with old age damage, and this is modern contamination that snuck into the sample. Only after all three of these layers, dense bone selection, sterile extraction, and careful computational reassembly, can researchers say with confidence that they are looking at a real signal from a 50,000-year-old skeleton, and not noise or contamination dressed up as history. For a long time, even with all of this technology available, Southeast Asia's heat and humidity remained a wall that few labs could get past for anything approaching 50,000 years of age. Successes elsewhere in the world kept piling up. Ancient genomes from cold parts of Europe and Siberia, some over 40,000 years old, were sequenced with increasing routine. But tropical successes stayed rare, small, and often limited to much younger remains, a few thousand years old at most. That gap is exactly why, when researchers eventually did manage to pull meaningful ancient genetic information connected to very old Southeast Asian human remains, it felt like watching someone unlock a door that the whole field had assumed was sealed shut. Now here's the myth. This video wants to take apart directly because it is one of the most common misunderstandings people carry about human history, and it deserves two passes instead of one. The first popular myth goes something like this. Modern humans walked out of Africa, and every other type of human, every other branch of our family tree, simply vanished before we got there, cleanly and completely replaced with no overlap. This idea, sometimes called strict replacement, pictures human history as a relay race with one baton, passed cleanly from one runner to the next, no crossover. It is a tidy story. It is also wrong at the level of detail that actually matters. Genetic evidence gathered over the last 2 decades, including Material connected to Southeast Asia and the wider region around it shows something messier and far more interesting: overlap. Modern humans moving through Asia did not always arrive to empty landscapes. In multiple regions, they arrived to find other human populations already living there, upright, tool-using, socially organized groups that had been established for tens of thousands of years before modern humans showed up. And in more than one case, these groups did not simply vanish without contact. They mixed. Interbreeding happened, and the genetic proof of that interbreeding is still sitting inside the DNA of people alive today, particularly in populations native to parts of Southeast Asia, New Guinea, and Australia, who carry small but measurable amounts of DNA inherited from an archaic human population that once lived in this region. The second myth worth confronting sits right next to the first one: the idea that any of this ancient mixing makes one modern population more pure or more evolved than another. This is a claim that has been used, historically and sometimes still today, to build false hierarchies between human groups. The actual science says the opposite of what that framing wants it to say. Every population studied, so far, carries some trace of ancient mixing with other human lineages, whether that trace comes from a Southeast Asian archaic group, a European archaic group, or another lineage entirely. Carrying more or less of a particular ancient DNA segment does not track with intelligence, moral worth, or any kind of ranking. It tracks with geography and history, nothing more. A population living in a region where a particular archaic group survived longest is simply more likely to carry a genetic trace of that specific group, the same way a family living near a river for centuries might carry local place names in their family history that a family from inland does not. It says something about where ancestors were, not who they were as people. So who were these other human groups that modern humans in Southeast Asia sometimes crossed paths with? And how do we know they were there at all if the environment destroys DNA so thoroughly? This is where the deep time zoom out comes in, and it needs more than one snapshot to make sense. Go back further than 50,000 years. In Southeast Asia and the wider region around it were not empty of humans waiting for modern humans to arrive. They were home to multiple different human lineages, cousins on our family tree who had left Africa in earlier waves, sometimes hundreds of thousands of years earlier, and adapted to local conditions over an enormous stretch of time. One of the most famous examples comes from the island of Flores in Indonesia, where researchers uncovered the remains of a strikingly small-bodied human relative, standing roughly a meter tall as an adult, with a brain much smaller than a modern human's, yet still capable of making stone tools and apparently surviving on that island for an extraordinarily long stretch of time, possibly overlapping with the earliest presence of modern humans in the wider region. This population shows just how much diversity existed within the human family tree at the same time modern humans were beginning to spread across the globe. It was not one human story unfolding. It was several, running in parallel, sometimes on the very same landmasses. Layered on top of that is a separate, genetically confirmed lineage, identified initially not from a full skeleton, but from a single small bone fragment found far to the north in a cold Siberian cave that turned out to belong to yet another archaic human population, distinct from Neanderthals, now known through their genetic signature far more than through their physical remains. Genetic detective work, comparing this population's DNA to people living today, revealed something remarkable. Their clearest genetic legacy is not concentrated in the cold region where that first bone fragment was found. It is concentrated instead in populations native to Southeast Asia, New Guinea, and Australia, at levels far higher than almost anywhere else on Earth. That distribution is itself a huge clue. It suggests this archaic population, or a closely related one, spent a significant part of its existence somewhere in or near Southeast Asia, encountering modern humans as they moved through the region on the long journey toward Australia and the Pacific Islands, tens of thousands of years ago. Two separate ancient timelines, one built from small-bodied island survivors, and one built almost entirely from genetic ghost signals. Both point to the same underlying truth. Southeast Asia was not a quiet hallway. It was a crossroads, crowded with different kinds of humans at once. Why does any of this matter beyond academic curiosity? Because these ancient contacts left real, measurable consequences in the bodies of people living today, and that is the human stakes beat of the story, carrying two connected threads instead of one. The first thread is about disease resistance. Some of the DNA segments inherited from these archaic populations are not just neutral, leftovers, sitting quietly and doing nothing. Researchers have linked certain inherited archaic gene variants to differences in how the immune system responds to particular pathogens, and to how the body processes certain environmental exposures. In some populations, these inherited variants appear to have been useful enough that natural selection preserved them over tens of thousands of years instead of letting them fade out randomly, which is a strong sign they provided some kind of survival advantage, whether that was better. Immune defense, better adaptation to local disease environments, or something researchers are still working to fully understand. In other words, some people alive today are carrying a small biological toolkit, quietly inherited from an ancient encounter between two different human populations in Southeast Asia, tens of thousands of years before written history began. The second thread is about migration and survival at a larger scale. The presence of archaic human DNA in populations native to Australia and New Guinea is tied directly to one of the most remarkable migrations in the human story. The journey of modern humans across open water using early watercraft to reach Australia at a time when it had never been connected to the Asian mainland by a land bridge. This means early modern humans in Southeast Asia were not just walking through forests and following coastlines. At some point, they were building boats or rafts capable of open water crossings, navigating without modern instruments and successfully colonizing an entire continent that no human had ever set foot on before. The archaic DNA carried by their descendants today is, in a sense, a genetic passport stamp from that journey. Physical proof that these ancient travelers had already been mixing with other human populations in Southeast Asia before they ever set out across the water. Now for the twist, the part of this story that even specialists did not fully expect, sitting right where interest in a story like this often starts to dip in the back half after the big reveal has already landed. Given everything covered so far about how hostile Southeast Asia's climate is to DNA survival, you would expect the region to have contributed almost nothing directly to the genetic record of these ancient contacts, only indirectly, through DNA found in far colder places and then reasoned backward. And for a long time, that expectation held. But newer extraction techniques, refined versions of the same dense bone, sterile lab, computational reassembly process described earlier, have slowly started pushing successful ancient DNA recovery into warmer, more difficult environments than researchers thought possible even a decade ago. Small, careful successes have chipped away at the old assumption that tropical ancient DNA older than about 10 or 15,000 years is essentially unreachable. Each success is modest, a partial genetic profile here, a confirmed identification there. Nothing as complete as the deep, rich genomes recovered from cold caves in Europe or Siberia. But each one chips further away at what was once treated as an absolute limit. The smaller, supporting oddity that reinforces this twist involves the sheer diversity uncovered on the physical, non-genetic side of the science, even where DNA itself has not survived. As researchers have mapped ancient skeletal remains across Southeast Asia and nearby islands more thoroughly, they have found body types and skeletal features so varied that they no longer fit neatly into a simple picture of one wave of modern humans passing through. Some skeletons show features that puzzle researchers, sitting somewhere between what is typically expected of fully modern humans and older, more archaic features, prompting ongoing debate about how much of that variation reflects normal human diversity and how much might reflect deeper, more complicated ancestry, mixing between more than two distinct human lineages rather than just one clean modern human meets one archaic group story. The truth is, researchers are still working this out, and that uncertainty itself is part of the honest picture, not a failure of the science. So where does all of this leave us once the twist has been laid out and the excitement has had a moment to settle? Here is what the evidence actually supports, stated plainly. Southeast Asia was home, tens of thousands of years ago, to more than one kind of human. Modern humans moving through the region sometimes encountered these other populations, and at least in the case of the archaic lineage best known through Siberian genetic evidence, that encounter included interbreeding, whose genetic traces survive today, concentrated most strongly in populations native to Southeast Asia, New Guinea, and Australia. Extracting reliable ancient DNA directly from 50,000-year-old bones in this hot, humid environment remains genuinely difficult, and researchers continue to push the boundaries of what is technically possible, chipping away at limits that once seemed absolute. Much of what we know about ancient interbreeding in this specific region has been reconstructed indirectly by reading the genetic legacy left behind in living populations and in ancient DNA recovered elsewhere, rather than from a single, complete 50,000-year-old genome pulled cleanly out of a Southeast Asian bone. Here is what the evidence does not support, and this matters just as much. This story does not support any idea that mixing with archaic humans makes one population more advanced, more primitive, or more pure than another. It does not support treating ancestry percentages as a measure of personal identity, character, intelligence, or worth. Ancient DNA can tell you a great deal about population history, migration routes, and even some aspects of biology, like immune function. It cannot tell you who a person is as an individual, what they are capable of, or where their personal destiny leads. Genetics describes populations and probabilities across huge spans of time. It does not describe you as a single person sitting here right now. If any single part of this story deserves to stick with you longer than the rest, let it be this. For most of human history, we were not alone, not in the sense of loneliness, but in the sense of company. Other human lineages walked, built tools, raised children, and in some cases crossed paths and mixed with the ancestors of people alive right now in a hot, wet, difficult landscape that later tried its hardest to erase every trace of their DNA. Some of that DNA still made it through, buried in bone dense enough to survive 50,000 years, and in the living genomes of descendants who carry a small, real physical link to a history far stranger and far more connected than the simple, one-wave story most of us grew up hearing. So go back to that same spot on your arm you pressed down on at the start of this video. Somewhere in Southeast Asia, in a case behind museum, glass or still sitting undisturbed in a cave floor, there's a bone from a body that lived and moved 50,000 years ago, carrying in some small fragile fraction of its structure a message that outlasted every flood, every fungus, every century of tropical heat that tried to erase it. That message did not just survive, it rewrote what we thought we knew. If a story like this is the reason you keep coming back to this channel, this is exactly the kind of evidence-first, no hierarchy, myth versus reality digging we build every video around. So consider sticking around for the next one, because the human story keeps turning up chapters like this one, hiding in bones nobody expected to talk at all. Thank you for watching. If this deep dive into ancient DNA and the hidden human history of Southeast Asia gave you something to think about, consider subscribing so the next chapter of this story finds you when it's ready.